My name is Rob Progar. I'm the National Program Lead for Entomology and Pathology Research for the USDA Forest Service. And I'll be presenting a series of initiatives and research conducted by the USDA Forest Service and its partners on pollination ecology. Several large pollinator data collection efforts are underway, and I cannot give them the level of recognition they deserve in a short presentation. But I encourage you to reach out to or invite them to future meetings. Starting with an initiative to collect baseline and effects monitoring information for native bees on 6 national forests surrounding the Great Lakes. The objectives of the project are to assess the impacts of management activities such as invasive species removal, prescribed fire, timber activity, or mowing within the previous 5 years on bee abundance and diversity. They collected more than 17,000 bees of 258 described species over 3 years from 2017 to 2019. Many species were identified as sensitive by the U.S. Forest Service and/or state listed. Some outcomes of the effort. They found that recent fire treatments increase bee diversity, and they are developing a bee rarity index and an eDNA protocol. Next is a new and interesting project on the development and effectiveness of pollinator habitat in the Minneapolis-St. Paul metropolitan area, or MSP. This study, led by Learman and Kay, could have significant and positive impacts on pollinator populations in the area. They are establishing a study testing the effectiveness of the new Minneapolis statewide bee lawn program for transforming areas within the MSP into refuges for pollinators. Starting in 2022, they will monitor bees in 80 residential yards scattered throughout the Minneapolis-St. Paul metro area. Yards include a range of management types from active bee lawns to passive management to high-intensity traditional lawns. Detailed information on our optimal soil properties, plant mixes, lawn size, and location for reporting pollinators will be collected. They will conduct citywide modeling to identify additional neighborhoods ripe for pollinator habitat. One goal is to understand the barriers for installing bee lawns and managing lawns for pollinators. Log landings are areas commonly subjected to soil compaction from log handling equipment. Poor regeneration and soils in highly compacted areas of national forests present opportunity acres for pollinator habitat. Pyle and others developed an integrated experiment that examined soil mitigation efforts such as ripping and incorporating biochar, planting native pollinator seed mixes, and evaluating subsequent bee habitat. In 2021, they monitored bees in 30 log landings in 3 national forests. Research and monitoring will inform best management practices, or BMPs, for Forest Service, state, and privately owned land to create pollinator habitat as a part of logging practices. They are collecting baseline data on bee species that occur in Midwestern national forests. They are assessing how silvicultural management practices affect bee communities forest-wide. They are also gaining an increased understanding of how soil amendments and which plant species contribute to wild bee habitat. A large collaborative project is currently underway in the Pacific Northwest between the USGS, U.S. Geological Survey, and Forest Service regional botanists and pollinator coordinators to collect environmental DNA for bees and other pollinators. The need for this work arose to better determine distribution and abundance of western bumblebee and other pollinators. Environmental DNA, or eDNA, has potential to improve the accuracy for pollinator community assessments. DNA left behind by arthropods on the surface of flowers during foraging is collected and preserved. Forest Service employees representing 14 national forests in the Pacific Northwest region collaborated to implement an eDNA collection protocol developed by partner researchers at the USGS. They sampled approximately 30 sites from montane, mid to high elevation meadows on Forest Service lands. Some of the outcome: the research is testing and optimizing pollinator eDNA collection methods. They are assessing the efficacy of using eDNA protocols to detect western bumblebee in the field. They also compare detection probabilities using single species detection methods such as PCR and a broad taxonomic identification approach or metabarcoding. Native bee monitoring on the Starkey Experimental Research Forest was established to gather knowledge about native bee communities of the inland Northwest and their potential responses to common land use practices on public lands, for example, riparian restoration and timber thinning to reduce fire risk. Rowland and DeBano conducted 2 projects assessing seasonal abundance and diversity of native bees and floral resources at the Starkey Experimental Forest and Range in Northeast Oregon. The first project assesses responses of native bees and floral resources to riparian restoration with varying levels of grazing pressure by wild and domestic ungulates. The second project assesses the effects of timber harvest on native bee habitat, abundance, and diversity. Some of the outcomes: more than 220 species or morphospecies of native bees were identified in 6 years of sampling in riparian areas. The effort greatly expands the knowledge of the seasonal bee community in forested rangelands. They document the occurrence of the western bumblebee, a rapidly declining and sensitive species used by wildlife programs on national forests in Oregon and Washington, and ongoing assessments and monitoring. Their data is shared with local national forests as well as providing specimens to guide future monitoring at districts and forest levels. Shrubs commonly planted in restoration projects such as willows provide important early foraging resources for bees before herbaceous blooms are abundant, offering an unexpected benefit of these ubiquitous plantings. Future restoration can use bee-plant relationships discovered through this study, including use of important non-native species, to guide restoration plantings for multiple benefits. The next project was designed to develop land management strategies such as prescribed fire to restore disturbance-dependent oak savannas and pine barrens, so whole site prescribed fires don't negatively impact refugia for bumblebees and other pollinators. Schutt and others used mark recapture, telemetry, tracking, nest boxes, pan trapping, and netting at 7 sites on National Forest System lands and 7 sites on Wisconsin state land in 2020 and 2021, when half the sites burned. The desired outcome is to improve conservation by characterizing bombus foraging and nesting habitat use before and after fires to inform land management recommendations. The next study arose from the 5-year drought in California from 2012 to 2016 that resulted in dramatic tree mortality across the central and southern Sierras. Fettig and Runyon are monitoring forest pollinators from 24 field sites across 4 national forests to determine changes in pollinator communities resulting from changes in forest structure and composition. This last project, titled Pollinator Partners Across Landscapes, or PALS, by Williams and others, is designed to restore lost native plant diversity caused by past management activities and competition from invasive species. This may be particularly important following thinning to create forest gaps intended to promote short-term pollinator habitat and long-term habitat for other important species such as the northern spotted owl, marbled murrelet, and Humboldt martin. Some knowledge gaps. We need to understand plant-pollinator relationships to inform development and successful deployment of native plant material mixtures that support the spectrum of potential pollinators in the Oregon and Washington Coast Range. We also require more information on how these mixtures will support the natural regeneration trajectory of trees to restore requisite old-growth forest structure. And that's all I have. Thank you for your attention.